everybody, and welcome to today's edition of the Short Pharmacity podcast. My name is Nels, and I'm going to be your host. I'm glad to have with me Diana Löber in the line today. She's Global Product Manager of Vials at Schott Pharma, and she will be sharing her knowledge about a very specific kind of vials, EVERIC Strong and EVERIC Smooth. Hi, Diana. Nice to have you here.
Hi, Niels.
Some of you may have right now a déjà vu, and this has a good reason. We hosted Diana already some time ago, and Diana explained to us about the two models of the EVERIC family, the EVERIC Strong and Smooth. So right away, my first question to you, Diana, why doing it again, or what is new?
So I can really say a lot is new and a lot has been done. So the last time I showed you the results of the line trial, we did in cooperation with Roche on a bulk filling line. And the most amazing result was that we could lower the final reject rate by 98% using EVERIC. In addition, we showed that there was no sticking and climbing of the vials, that handling was much easier, and that the outer coating actually protected the strength of the vials. But of course, lowering the final reject rate has the biggest leverage. Just think about the money that can be saved if you do not have to throw away your filled product in the end. Yeah, and since then, we continued working closely together with our customers. And of course, within these cooperations, additional questions came up. So we took these questions and worked closely with our R&D department, with our manufacturing sites, and also with partners to get the answers for our customers.
Stop, Diana. So could you maybe go one step back and quickly let us know what EVERIC Strong and Smooth actually is? I guess not everybody hearing this podcast has taken part in the past Pharmacity session.
Sorry, of course. So EVERIC Strong and Smooth consists of two combinable modules, strong and smooth. Let's start with the strong module. And as the name already explains, it's all about its optimized strength. When we started the project, there were actually three main restrictions for our R&D back then that we defined in our user requirement specification. So first, no change of class composition. Second, no additional chemical treatments. Third, stay within isogeometry.
Why did you set yourself these three restrictions?
So let's first have a look on the first restriction, no change of class composition. So borosilicate glass has been proven to be a safe and reliable primary packaging material with a long track record. It perfectly found, let's say, the sweet spot between processability on the one hand and chemical resistance on the other. Other glass types, as for example, aluminosilicate glass, actually need additional treatments to achieve the hydrolytical resistance level required by authorities. And this is why we defined in the URS no change of class composition. Regarding the second restriction, treatments, for example, chemical toughening, of course, lead to great strength levels, but you do not want to be there if a toughened vial breaks. It bursts literally into a million pieces. And I do not want to be the one who has to clean up the filling line afterwards. And in addition, toughening makes the vials so hard that parts of the fill and finish machine are damaged, leading to higher wear and tear and therefore higher costs in the end. And coming to the third point, ISO. I think that's quite obvious. The ISO standard gets more and more adopted. Especially our large customers shift their portfolio whenever possible towards ISO to reduce complexity. So the ISO geometry is really satisfactory, and we do also all our innovations based on ISO to make sure its compatibility with adaptive or ready-to-use platform, but also to provide early sampling in small quantities from stock.
Okay, I understood. Thanks for the declarations. So that is regarding the first module, the EVERIC Strong. What is now EVERIC Smooth? What is the relationship to strong, and why should you have this if the vials are already so strong?
I mean, of course, we can produce a geometrically optimized, flawless vial on our line with no glass-to-glass contact, a cosmetically improved glass tube, and an automatic backend. But if you do bulk filling, the vials will necessarily experience unavoidable contact on the fill and finish line, leading to cosmetic defects that might decrease the vial strength and increase final reject rates. And this is why our approach is take the vial with the optimized strength, EVERIC Strong, and combine it with an outer coating that protects its strength, EVERIC Smooth.
Okay, the sounds for me, EVERIC Smooth is only there to protect EVERIC Strong?
No, by far not. So actually, EVERIC Smooth was developed to avoid sticking and climbing of vials from the filling line. And this especially occurs after the deprogenation oven, when the vial's outer surface is unsaturated and therefore rough, but also very vulnerable. So the secret behind EVERIC Smooth is that the outer coating has a more than 50% lowered coefficient of friction compared to standard vial, making it more slippy. And you can really feel the difference when you rub two vials against each other. And another advantage of EVERIC Smooth is that you can increase the line speed because the vials run smoother through the line. And this we have proven in cooperation with Optima. And they accomplished to create a test machine setup with a possible filling line speed of up to 750 vials per minute. And they tested our vials. And I can tell you, I've been there when we did the trial, and it was indeed hard to see the vials anymore.
I can imagine impressive speeds. You've mentioned just right now that the sleepy surface or that sleepiness, isn't it a challenge to then put on the label on the vial?
Yeah, so this is one thing we also tackled during the last year. So we worked together with UPM Raffertag, and they came up with a solution here. And of course, it's actually not about the label itself, it's rather about the adhesive needs to work with the label and the coated vial. And in addition, there's another label manufacturer who managed to provide a solution here, and probably a third label provider will also be tested. And maybe some words regarding the slippiness in general. This was, of course, the challenge that we well conquered here, finding the right coating material with the right properties. And our R&D department started with over 300 possible compounds, and we ended up in the end with 2 that perfectly suited our purpose and fulfilled our tight requirements.
Okay, 2? You mean that there are actually 2 EVERIC Smooth options?
Yeah, indeed, there are 2 options. So the first one is a silicone, and silicone is a well-known cladding agent in the pharmaceutical industry. And for those customers who are okay with using silicone, this is a suitable option. However, there are also customers who do not want to have silicone in their facilities at all. And mostly this is due to very high value drugs they are filling. Often these kind of drugs, for example, monoclonal antibodies, are really sensitive, often towards silicone also. And for these customers, we do have an innovative next generation cladding agent, silane. And it is a monolayer coating that covalently bonds to the surface and is even thinner than silicone. And a colleague who is an expert for coating, he, I think, drew a nice mental picture, because he said that the coating it's like an accordion, like it has an accordion structure that acts like a damper on the outside of the vial. And both coatings are, by the way, hydrophobic.
Okay, thank you. Can you provide us some details? How does the process work to get the coating onto the vials?
Yeah, so this is a really good question, because now I'm also allowed to disclose more information on that topic. And the Pharmaversity session also has a focus on that part. So you get the details in the presentation, but I can, of course, share the most important facts with you as well. So, of course, we investigated different coating process possibilities. And after carefully evaluating different options, we deliberately decided against a spray coating option and in favor of a dip coating process. So why is that? I mean, the worst case scenario for our customers probably is to contaminate their drug product. So we needed to absolutely make sure that no contamination takes place due to EVERIC Smooth outer coating. The process, so meaning the application of the coating, therefore, of course, represents the most critical part.
What is exactly the issue with the spray coating, Diana?
Yeah, so actually, I think there's even not that much explanation necessary. So just think of a kind of spray that you use in your everyday life, like nasal spray, hairspray, glue spray, etc. So let's take hairspray as an example. You can go really close to your hair, and then you get a spot with a lot of spray on it, followed by surrounding areas with a thinner layer. And when I want to get volume in my hair, I usually put the hairspray on from more distance to cover a larger area. And what usually happens, but maybe Niels, you're better with this, and this is just my problem, but what usually happens with me is that it feels like my whole bathroom bathroom is covered with a layer of hairspray. So what do we see here? So first, it's very difficult to get a homogeneous distribution with spray. Second, it is very difficult to avoid contamination of the surroundings by the spray mist.
I can only confirm this situation. Understood. Thanks for this very visual and clear explanation. But also deep coating poses a potential risk. Like, for example, if I would dip too deep, how do you make sure that no contamination takes place?
So maybe I have to go one step back and explain that there are actually two possible kinds of contamination. So the first one is direct contamination, meaning that the coating solution itself gets in contact with the inner surface of the vial. And the second contamination possibility are actually volatile components. And these volatile components might occur, for example, due to an increased temperature, and then might contaminate the vial from the inside. So what do we now actually do to avoid any kind of contamination? So we have a special design gripper system that makes sure that the vials are safely closed during the dip coating procedure. And in addition, there's a mechanical limitation in place, avoiding that the vial can be coated more than up to the shoulder. And this also avoids that the handling system gets in direct contact with the coating solution, because this could then be a source of recontamination as well. We, of course, also investigated if there could be contamination by these volatile components. And therefore, headspace analysis have been performed at different temperatures, up to 220 degrees Celsius. And this is actually the limit of the testing method. And we could show that there are no volatile components that might lead to contamination, neither of the vial nor of the handling equipment.
Thank you, I understood. So now the coating is on the vial. You just mentioned a moment before that the biggest fear of the pharma companies is the contamination of the drug. How do you know that the coating doesn't come off and get into the drug?
So we made many tests here to prove this for both coating options. And just to give you some examples. So we performed scratch tests to show that no particles generate even with increasing forces. We weighed the vials before and after deprotonation to make sure that no coating got off. We checked the water before and after the processing during our line trial with Hopper and Roche to prove that no contamination took place. And in addition, our colleagues from Schott Pharma Services performed so-called carryover studies to check if the coating might migrate into the vial via time. And this happened under harsh accelerated conditions. So vials were incubated for 6 weeks at 60 degrees Celsius, and we could show no contamination took place. And as you can see, we take the concerns of our customers really seriously here. And in the end, about patient safety.
Sure. Now, maybe coming to an end, Diana, one general question what I ask myself. So here we talk about bulk primary packaging, bulk vials. So big quantities, high-speed filling lines. At the same time, we see and hear that future goes direction ready to use. So less quantities, dedicated, these flexible filling concepts. So why still innovating in bulk vials?
Of course, you're right. So the future is ready to use. We see more and more customers adopting it, especially for smaller quantities, but also many want to shift their whole production towards ready to use in the long term. And that's actually the crucial point, long term. Yeah. So the change from bulk to ready to use will not be done in 1 decade. And in my opinion, there are also drugs which will probably never change to ready to use. And with the pandemic and the resulting unexpected jump in demand for mass vaccination, we all just saw that improving line efficiency for bulk is maybe still more relevant than ever.
You're right. Okay, so Diana, thanks a lot for taking the time and sharing with us experiences and the results of the developments done within the EVERIC family. I'm sure we will have the pleasure to host you again here in future and learn more about EVERIC or the other very interesting topics you are busy with. Thanks also to the audience for hearing this podcast. Take care and bye-bye.
Bye-bye.